So today, I think it's fair to say that we're lucky to have Jose Manuel Alonso here to visit and deliver a talk. He's a distinguished professor at the State University of New York, colleagues with some other good friends of ours up there. I think he's best known for some real foundational work teasing apart the properties of on and off pathways in the primate visual system. And part of the reason I've followed his work for many years is that he's not just interested in the pathways for their own sake, but how they are related to the processing of different information sources, all of which are tied into the perception of depth and motion. So he's studying currently how the on and off pathways respond in terms to different levels of focus error and how defocusing images on the back of the eye can relate to the development of myopia and amblyopia, how the on and off pathways are related to that. Has examined how on and off pathways relate to and influence the development of binocular receptive fields. And for those of you who know me, I'm very interested in binocular processing and stereopsis. He's also done a lot of important work understanding how all this relates to motion processing. So I have been a bit out of touch with his very most recent work over the last couple of years. So I'm very excited to get caught up to date. And I hope you'll all join me in welcoming Dr. Alonso.
Thank you. Thank you so much, Johannes, for inviting me here. It's really a pleasure to be in and get to talk to some old friends and some new friends that I haven't seen not so long ago. So this is kind of an interesting talk for me because what I'm trying to do is to create a bridge between the work that I have been doing for a long time in on and off pathways. I'm trying to understand how neurons respond to light and dark stimuli, receptive fields, cortical maps, and bridge these to my new interest, that is myopia, nearsightedness. And I will try to show you that all these basic things that we know, this low-level vision about on and off pathways, about luminance increments and decrements, can teach us a lot about perception and about vision in this disease that is becoming a world epidemic that we call myopia. So I'm going to start. Let me pass the first slide. With this is one of my favorite slides. This is from Javier de Felipe, one of the best neuroanatomists in Spain, that shows this structure that many of us love to study. That is the brain. And I bring it here because it makes a very important point for my talk. It's just, the brain can many different sizes, many different shapes, from the huge one of the human right here to the tiny dot that you see of the mouse. And it could even, Javier de Felipe here just showing mammals, but they could include there a fly that you wouldn't be able to see because the size would be so small. And all of these brains have something in common. All of them can process visual images, and because they can process visual images, they all have on and off pathways, all the way from the fly to the human. So on and off pathways are very, very popular in vision. And what do they do exactly, and how and why do we need on and off pathways at all? Okay, so and one thing that I like to, of course, in vision, we need to process light stimuli and dark background, like this one. It could be a star in the night sky, and dark stimuli and bright background. It could be a fly against a blue sky. Okay, you can think also in terms of temporal changes. Just something that becomes very bright and something that becomes very dark. Something that I really want to make emphasis here is that on and off is also popular across different sensory systems. You have also on and off in the olfactory system, for example, and it works pretty much like a vision. You can imagine like a background smell of roses, for example, and you can, if you approach the roses, you're going to get an increment in the smell. If you get far, you're going to get a decrement. This is all on and off pathway. So it's something that is also very, very prevalent across sensory systems. And I'm going to leave the question of why do we need on and off pathways for a little bit later. But first, let me tell you something that has been, one of the main things that we have been trying to communicate over the past 10 years coming from my lab. And it's this idea that, that it was very prevalent in the vision community, actually, that, we have on and off pathways that is amazing. They are on and off pathways originate at the very first synapse of the visual system, okay? You have the photoreceptor, the light sensor, and the first thing that you do, doesn't matter if you're a fly or if you're a human or if you're something, a carnivore or a primate, is to split the signal into on and off. And if we go to the olfactory system, the example that I'm going to give you, exactly the same. Actually, from the sensor, you already are splitting the signal in on and off. And the idea that was there, it's still maybe in textbooks for a long time, is that you go through this big effort, okay, that this first thing that you do is you split the signal, you keep on and off pathways segregated in the retina. You go to the LGN, and depending on the species, if you're a ferret, you are still segregated in layers. If you are any other visual animal, you are still segregated at the level of single cells, so that some cells are on center, respond to light, others are off center, respond to dark. But then when you get to the cortex, the main idea is that you form Gabor filters, complex cells, phase independence, and then on and off is gone. That was the idea. And that's an idea that over the years, we have made work to support the idea that this is not true, actually, that the on and off pathways remain segregated in primary visual cortex and through the visual cortex. And this is an idea that I want to summarize a lot of work, but in this slide, this is based on work from carnivores, and I always like to show my half orange, just to remind everybody that with this number, 0.5, you can remember most of the work that we did about on and off segregation in the primary visual cortex. You have 0.5 millimeters to go from an ocular dominance column from the left eye to the right eye. You have 0.5 millimeters to go in the opposite axis from an on domain to an off domain. The receptive fields actually move at 0.5 receptive fields per millimeter. And with this, you have a structure in the cortex, okay, where you preserve this on and off segregation that happens at the very first synapse of the visual system, so that you have on domains, off domains, just as you have left and right eye. This is an important idea that I need to put here before I move forward into the other things that I'm going to tell you, because it brings this idea of segregation and functional specialization on and off pathways. We think about on and off pathways just in the same way we think about left and right eye. Left and right eye, also in the primary visual cortex, they combine, they form binocular receptive fields, but they can still operate independently. You close one eye, you can see. We think that it's the same thing with on and off pathways. You lose an on pathway. In fact, there are people out there without on pathways. Just a mutation, a small mutation at the level of the connection between the sensor, the photoreceptor, and the on bipolar cells, and you lose the entire on pathway. And these people are not blind. They can see perfectly. If it is a bright day, outdoors, they have problems seeing in dark backgrounds, and they have problems seeing at low light. They have night blindness because the entire rod system, your entire rod system, is also part of the on pathway. So we have this idea that on and off pathways remain segregated in the cortex, and this is part of the work that all these papers that I show you there. And because they remain segregated in perception. Also, your perception of dark and light stimuli are going to be different, and it's going to have different properties. So on and off segregation in the retina, in the thalamus, in the cortex, and beyond. And with this idea of on-off segregation, we put a theory of how cortical maps form, and it's a very simple idea. And this is the last thing I will tell you about the cortex. From here on, I'll go into on and off, and I will tell you why the cortex is important for that. So the main idea is this, is that as you get to your brain grows from the mouse to the human, and you have more real estate for each point of visual space, then you have the luxury of start segregating things. If you have, like in a cat, for example, 2 millimeters by 2 millimeters representing the same point of visual space, you can put, sorry, 1 millimeter by 1 millimeter. You can put 0.5 for the left eye, 0.5 for the right eye, on and off. And then from this segregation, this is the main idea that we have in this paper, you start creating orientation columns, ocular dominance columns, and on and off domains. But here, when we got to this point, we started saying that this, in some way, this was the beginning of the bridge towards the myopia story that I'm going to tell you. We started seeing not only that on and off were segregated, but they were different. And this was not new. As I will tell you, if you go back, we knew this. But we were seeing things that these differences that we thought could have implications on how we see light and dark stimuli. And I use this terminology, light and dark, on and off, vision through on and off pathways has been described in many different ways. You will see it like increments of lights or decrements of lights. You could say like light and bright, brights and darks. We like light and dark because it comes back to one of my favorite studies about light and darkness that comes from Leonardo da Vinci that used, he got like 6 different books about difference between light and dark. So this is the terminology that I will be using. Light is something that is brighter than the background, and dark is something that is darker than the background. So when we started looking at the cortex, we started realizing that there were many differences. There were differences in the speed. For example, the off pathway was faster. And in some way, it makes sense because if you go back to the retina, the receptors, the off pathway moves through an inotropic receptor that is fast, where the on goes through a metabotropic receptor. This is in mammals, that is much slower. Resolution, for example, we saw in the cortex there was much finer retinotopy resolution for off than off. But there was one difference that really was the most important for what we were going to do after, and it was contrast sensitivity. And it's something that I'm going to be telling you about a lot. So the cortex, what we started seeing in the cortex was something that in some way was very similar to what we were seeing in the retina. So in some way, we started thinking about the cortex as a way to study on and off pathways that they are in the retina, because they are already transmitting these properties very, very reliably. And this is, I always like to show this story. That is the thalamocortical scaling law. This is a beautiful paper from Chuck Stevens in Nature, showing that the number of LGN cells and the number of cells in the thalamus is strongly correlated with the size of area V1. And you can show the same thing across different species. Basically, as you start getting more retinal ganglion cells and more thalamic cells, you start making your cortex larger, and then you start being able to segregate things into on and off. As you think about the cortex of on and off domains, the things that we see about, for example, differences in contrast sensitivity, difference in the spatial resolution, are very similar to things that we can measure in the thalamus and the things that others measure in the retina. And I'm going to tell you mostly about 2 main differences that we find between on and off. And I start with the contrast sensitivity. The contrast sensitivity is higher for on pathways than on pathways. And this, you can see it already here. This is work from the DEM lab, single cell in guinea pig, where you can see that at low contrasts, the on is responding stronger than the off. And then at the highest contrast, the off is responding stronger than the on. If you measure, we have more recent measurements of humans, electroretinogram, and you see exactly the same thing. You see a low contrast, on responding better than off, higher contrast, sometimes, there is a strong response from off than on. We see this also in the primary visual cortex of primates. We see it in the primary visual cortex of humans with human VEP, and we see it in natural images. So if you take a natural image, any natural image, and you divide it by the median, and then you take the contrast, the density of contrast, and that are brighter than the median or lower than the median, you see something also very similar. We have more low contrast for light and dark stimuli. And this would explain why maybe so well preserved this difference in contrast sensitivity between on and off. So because we have more low contrasts for light and dark. When you look at humans, this difference in contrast sensitivity, just measured with a human ERG, just really very, very simple wires that you put in the eye and you just measure the B wave. There is some subtleties about how you measure on and off, but it's something very, very consistent. Each of these dots is a human subject, and the C50 basically is a measure of contrast sensitivity for darks. So it's the contrast that you need to reach 50% of the maximum response for darks, and the C50 would be the same for lights. If the value is lower, it means that you have higher contrast sensitivity. And every single subject, the contrast sensitivity is higher for on than off. And this is now in the retina, but the cortex would be the same thing. Is this scaling law where some of these very low-level properties, they are being just reliably transmitted. There is something, and here is where I'm beginning to make the bridge with the myopia study. There's another idea that was there in the vision community. Yeah.
What is each dot in that plot on the left?
In the left is a human subject, an ERG. So you measure the B wave, the amplitude of the B wave, and then you plot contrasts, and you get the contrast that generate 50% of the maximum response. Then you do the same thing for the D wave. The D wave is a different way to get the D wave because, but I can explain if you want. Basically, it's responses in an ERG for lights and darks. So an important concept that I want to bring, and this we think is important for myopia, is that there is this idea, okay, that contrast is independent of luminance, okay? And this is a very, very good idea when you're studying vision in a lab, because, hey, if you have a monitor that can only reach 50 candles per square meter, that's fine because you can, contrast is, doesn't matter if you're measuring with one that is 200, or if one that now is 1000. But this doesn't make too much sense, isn't it? Because particularly if you think in myopia, when you start hearing my colleagues talk about myopia, how important is being outdoors, probably one of the best things that you can do to reduce your myopia risk. And they really don't know what it is, but one of the basic things could be just brightness, you know, how bright the outdoor scene is. One thing that we find is that when we start using 1000 candles per square meter with these bright monitors that are now available, is that that contrast does change. The contrast response function changes with luminance. And what is cool is that it changes differently for on and off. For on, this contrast sensitivity becomes even more pronounced and you saturate more. And for off, it becomes more linear. So if you get a ratio of the C50 contrast sensitivity from C50 and for dark and light, this ratio increases as the image becomes brighter. A way to illustrate this, there's a lot of work that goes there, but a way to illustrate this very clearly is just with a cartoon, okay? Imagine that you are indoors. This is the contrast response function for the on pathway, okay, where you can distinguish an F from a P. The P is a little bit brighter than the F. You can also distinguish a dark F from a P dark because, you know, the function is not saturating a lot. But when you get outdoors, when you're in a beach scene, what is going to happen is that your off contrast response function now becomes extremely linear, and the on saturates a lot. This is, in some way, you know, it's not news. This is the Weber law, which, again, is true with any sensory system. As you make the, you know, stimulus stronger and stronger, you need bigger differences, and eventually you saturate. But I think that it has important implications for vision. First, you know, it explains when you are at the beach, in a very brightly illuminated, you're going to have a very, very good way to discriminate the eye line, for example, expressions in a face, okay, because you have this linearity in the off pathway. But you wouldn't be able to tell me if a reflection in the ocean, for example, these very bright reflections for the sun, one is brighter than the other because you are in this saturating function. And then you cannot tell, hey, this is brighter or not. And it doesn't matter. It matters much more the darks than the brights. This is going to be very important because what you are seeing here is that as something gets brighter, you are going to activate the on pathways much, much stronger. And this is something that you see with an ERG. Clinicians have known this for decades. They know that if you have a pupil dilated, you're going to get a very, very strong big wave and this is why they have to dilate the pupil to get a good ERG. So the intensity, not just the contrast, the brightness of the stimulus is very important to drive on pathways. One more thing about difference between on and off. We have many. These two that I'm telling you, I think that they are the most relevant for the story that I'm going to tell you after. And it's peripheral vision. So for this, we left our electrophysiology. We start using these head-mounted visual displays. We have an eye tracker. So we can ask, and we have this noise there that you see, similar to this. We ask the subjects to fixate at the center. And now we can present the stimuli in 90 different positions in the periphery and can be light or dark. So that's how we measure on and off. And then we ask them just to tell us, we have different versions of it. In the simplest one is they have two clickers and you tell me it's on the right or it's on the left. In the most recent one, we have the eye tracker actually telling us, where, move the eye to the target. And then with this, we can measure reaction. We can measure reaction time also with the clickers, but each one has advantages and disadvantages. This is just, to show that we measure the eyes and, you have also accelerometers. You can see where the head is going. But the important thing is that you can start saying, hey, let's play with contrast. Contrast is something that we are getting very interested in. And of course, you increase the contrast. And, if you plot percentage of errors, how many times do I detect? You say left when it's left or right when it's right. Percentage of errors, you have more errors with low contrast. As you increase the contrast, you have less errors. But what is interesting for us is that there is a difference between if the target is light or if the target is dark. And here it seems very small difference. You see, here you are making more errors with the blue than with the red, which is what you would expect from an on pathway that has higher contrast sensitivity than an off pathway. And here you are making less errors with the blue than with the red, which is maybe related with the fact that, at high contrast, the dark targets, in the ERG, what I showed you, you're getting a strong response from the off than the on. Here, if you see this, you say, Jose Manuel is pushing. This is a tiny difference there. But now, if you start looking at the correlations, you say, whoa, if I plot contrast against the LiDAR ratio, I get a very, very strong correlation. And it doesn't matter at which eccentricity I look. So as I increase contrast, I make, remember, the errors are going down. It's not that I'm making more errors. I'm just making relative more errors with light stimuli than dark stimuli as increased. I'm getting better with darks at high contrast. And as I go to low contrast, I get a little bit better with lights. And you can make the same trick with eccentricity. You can say, hey, what happened if we fix contrast and now I increase eccentricity? And there is also a correlation, not as strong with the contrast. Contrast is more important than eccentricity. But also, as you get farther and farther from your point of vision, you start making more errors with light and dark stimuli. You can make a game with this and say, hey, who is better and who gives me the best goodness of fit? And the answer is that if you do it with eccentricity, not very good, you are around 0.2, you do it on contrast much better. And if you combine both, actually, you're doing pretty well. So with this fit, you can predict or find a correlation with the 48 possible combinations of eccentricity and contrast. The important thing here is that as I increase contrast, I make less errors with darks than with lights. As I increase eccentricity, I make more errors with lights than with darks. So you put it in a different way. I mean, if you are looking far and there is something that comes from your peripheral vision, you will do a better job at detecting it if this is darker than the background, what we call dark, than if it is lighter than the background that we call light. So now I'm at the middle of the talk, half an hour, and I'm going to move from these on-off basics that I kept to the bare bones, okay? how it came from, very popular across the animal kingdom. Everybody that has a retinal image needs on and off. Popular also across sensory systems. There is something really interesting about what is brighter than the background, or what is more intense than the background, what is less intense. We left this question about why do we need it open, but maybe the answer is just because they are different. They are very different. It's not that, one is a negative version of the other. It's just they are different. They have different control sensitivity, they have different speed, they have different spatial resolution, and probably they are different because nature is different. Outside, in our natural scenes, lights and darks have different statistics, and the contrast may be an important one of them. And of course, the brightness is going to be another important. The on is going to be much better at telling us, hey, this is getting really, really bright, and I'm saturating a lot because now I'm being very, very strongly activated. So we spent a lot of time. This is 2019, yeah. Around 2019, okay, we were all very interested in this difference between on and off pathways because we started thinking that it could have implications in vision, because we had this belief that in cortex they were on and off pathways were also segregated. There was this functional specialization, all these things. We thought that we could see this idea that the on and off combine, and light and dark stimuli doesn't tell you, on and off function. We didn't think that this was right. So we could use actually lights and darks to measure on and off function. And again, when you look back in the literature, you always find somebody that said that before. Munson and Schiller, for example, they showed long time ago that if you block with APB the on pathway in a macaque, the macaque is doing great at detecting dark stimuli, but for bright stimuli, you have to make it really, really bright for them to tell you there is a stimuli in the screen. So, but here, when we started looking at the differences, I started seeing a coincidence. It was too much of a coincidence to ignore it. And it's all the things that the on pathway didn't like are the things that my colleagues in optometry were telling me that there were risk factors in myopia. And in fact, we started with amblyopia. This is why this cute, the son of Jean, a member of my lab, is telling us here, hurry up, because really, really, we can solve this. And, and we are not getting younger. So he doesn't have amblyopia. He just put a patch just for us for the picture. But we started with amblyopia because, we were very obsessed with the cortex. And we make this prediction. I'm not going to go, it used to be a big part of my talk, but not. And based on this on and off difference that we see, particularly in spatial frequency, we predicted that amblyopia not only should make your lazy eye weaker, but also should make the on pathway weaker than the off pathway. And we found a strong, confirmation of this prediction. Not only we found that it makes it weaker, but actually we found a correlation between the severity of amblyopia and this on-off asymmetry in detection. So in other words, to put it simpler, if you are amblyope, you have a much harder time seeing light stimuli on noisy background than dark stimuli on noisy background. And if you are very amblyope, you won't be able to tell me to count the number of light targets in white noise, so embedded in white noise. So we were very, very happy when we confirmed our prediction in amblyopia, but we started thinking, hey, maybe the same thing is happening in myopia. Amblyopia and myopia could be the two sides of the same coin. So in amblyopia, you're getting a non-weak pathway in the cortex, but in the retina, in myopia, you're getting the same thing in the retina. So the advantage of myopia is that, half of the world, almost, and it's getting worse. I mean, just if you are not familiar with this topic, just to give you an idea of how big this problem is, if you are 19-year-old in Singapore, you have a 95% chance of having myopia. Myopia is huge. And what drives me crazy, okay, is that it's a much simpler problem than anything that we are thinking about cognitive function. It's a simple problem, okay? We know that environment is important. We know, it has all the elements that we love about plasticity, about structure and function, about how the environment shapes the brain. All of this you get in myopia. In myopia, what is happening, okay, is that something that you are doing, the genes are important. I told you about, let me make a pause here, because this is important to say. if you have a knockout completely, and there are humans with that, of on pathway, and I tell you, it's just a synapse, a mutation in the synapse between the sensor and the bipolar. Not only you get night blindness, you get very, very high myopia. So there is something about the on pathway and myopia that was already there. And then you have the work of Michel Pardieu, for example, using knockouts in mice, showing also that if you knock out the on pathway, you have more, you are more susceptible to myopia in mice. So there were all these things there, but then from the same, in some way, the amblyopia and myopia have been related from the very beginning, because probably the first most impactful paper in myopia was from Raviola and Wiesel, that they noticed that they were, closing one eye to cause amblyopia. And then they noticed that the eye was getting also huge, very, very long. And this is how they discovered, hey, visual deprivation causes myopia in macaques. So there was this thing there also, this relation between the on pathway. But from the work, these asymmetries that we were seeing, we predicted that the on pathway should be weaker in myopes. And we asked, how can we check that? Okay, we did it in the amblyopes. It was very good because, usually amblyopes have an amblyopic eye and a fellow eye. Then we did our psychophysical test and we could see that, the amblyop was doing worse with light stimuli than the fellow eye. That was nice. But how could we do it for myopia? The ERG, the electroretinogram, that, one of the Nobel Prizes in visual neuroscience from Granit. But then, it got a little bit unpopular with the time, and now it's gaining popularity again. I think that it's a wonderful technique to measure non-invasively on and off pathways. And what do we find? We find that if you, and now the beauty of the myopia, okay, is that you can measure eye axial length. And it's a very, very accurate measure to the micron, okay? I put you in front of these machines that they're getting better and better, and I tell you exactly what your axial length is. Mine is, I won't tell you. I have a lot of myopia. Well, it's 27 and 28. The normal is 23. I have a very long eye. But if we put in the x-axis, okay, different axial lengths, and then we measure, let me start with the bottom one. We measure the response to 30-40% contrast, you find a correlation. As the axial length increases, the response to this intermediate contrast decreases. It's a significant correlation that you don't find in the off pathway. And surprise, this we didn't predict. If you look at the latency, the off pathway doesn't change, but the on pathway is becoming slower.
So I presume this is all properly focused.
All properly focused. Yeah, good point. All properly focused, but in some way, it wouldn't matter because all of this is flash ERG. So you just put a flash and then in a dome, and then it's a full field ERG. Full field ERG. And this is kind of important for something that I'm going to, so thank you for that, for something that I'm going to tell you after. Full field ERG, no, nothing. So you just stare there in a dome. So we want to stimulate your entire retina, and we put, basically, I take the opportunity to tell you what exactly is the paradigm. You have a 500, so it's very bright, a 500 background, and then I give you a flash of 200 milliseconds, and it can be, 600 all the way to 1,000, or it can be, that would be my light stimuli, my own pathway, or it goes from 500 all the way to 0 or to 10, and this would be my off pathway. And this is important. This is the type of thing that is skipped before. And part of me wants to talk about it because it's different between the onset. In order to get these measurements, you really have to look at the onset and not the rebound. But I'm gonna skip that in order to get to what I think is more important. We are finding in myopia a correlation that indicates that the on pathway is getting weaker and slower. And this was an important thing for us because from this point, this is 2024, we started thinking more along the terms of, my entire career is about vision. Okay, you present a stimulus, you get a response in a neuron, or you get a response in a human. I never thought about motor, But when you start seeing this, you have to think about motor. Because and here I'm getting close to Josh's work, I have to think about the pupil, because it happens to be, and now, but I'm thinking about much lower level stuff than you do. So I'm just thinking of pupil light response, okay? The pupil light response, okay, you have a flash and your pupil becomes smaller, it constricts, and is mediated purely by the on pathway. Okay, you lose the on pathway, is complicated, it can go through the photoreceptors, it can go through the melanopsin cell, but it's on pathway, purely on pathway. So we say, hey, if you have a weaker and a slower on pathway, you should have a weaker and a slower pupil. And that's what we found. And this is important, but we think it's important for myopia. We have done lots of different analyses. There is a huge figure with, I think, the 10 panels in this paper in the 2024. But I like this one to show this one. If you get a pupil constriction of 3 millimeters, okay, it's going to take you longer to constrict than if you get a pupil constriction of 4 millimeters. There is a correlation in the peak time of the pupil constriction and the maximum pupil constriction that you get. In black is the controls. So these are people that don't have myopia. And in red are the myopes. And now what we separate is what they call low myopia and high myopia. High myopia is anything 6 diopters or higher. And you see that you already can see the separation in the low myopia and becomes bigger in the high myopia. So basically what it's telling me this is that the on pathway is getting weaker and slower in myopia, and your pupil is also getting weaker at slower at constricting with light. So think what this means. This means that when you go outdoors, okay, that is supposed to be one of the best things that you can do to protect yourself from myopia. If you already have it, you have a sluggish pupil light response. Maybe you're not going to take advantage of this brightness because you're not looking up to the sky. It's very bright. Your pupil doesn't constrict enough. It's slow doing it. So maybe you're outdoors, but you are looking at luminances, at radiance that are maybe equivalent to what you get indoors, and you're not getting the benefit of being outdoors. Just to, if you forget about everything I said, at least don't forget about this picture. Okay, this is just 2 people from 2 subjects, one with very high myopia, the other is with emetropia, in a 500 candles background, and then the pupil is very small, protecting the retina from the bright light, but it's a little bit larger in the high myopia. From all the thinking that we have now going on in myopia, we think that the pupil is going to be at the center of it for multiple reasons. I think that I will tell you one reason as we go. Let me move on. I'm going to make like a small detour from this visuomotor thing that we think is very important, because we put a lot of work on this paper, and it's important. We asked how on and off pathways, of course, when we were thinking about all of this, how on and off pathways are affected by the focus. That is, what you get when you get out of focus. It could be with a minus lens or a plus lens, and by magnification. That is something that when you put your glasses on, there are 2 things that you're affecting. You're affecting your focus, but you're affecting also the size of the stimulus. This, I'm putting an extreme example. If this is a 20 diopter plus, that you magnifies this grid, and then a minus 20 that minifies it. It's going to be a very brief tour, but worth it. So this is again in the cortex, but again, the main idea is that what we are measuring in the cortex is what you would measure in the retina. The advantage of doing it in the cortex is that we can do it in vivo. Okay, this is from animal models. And what we find is that if you are on, this would be the on-receptive field in red at the top, as when you start introducing the focus, it doesn't matter if it is minus 10 or plus 10, your receptive fields get larger. You lose resolution, your receptive fields get larger. But if you are an off, then the story is very different. Actually, your receptive field measurements, and this is very important, start getting smaller with the plus, because you're responding to magnification. And we know that this is magnification because we did it with spectacles and with contact lenses, and with contact lenses, that magnification is negligible. What you see is that the off shrinks with the focus, and the on expands. So it's really, really just a brief story, but I think it's important. This could be the average of all the cells that we did. if you use spectacles to cause the focus, the off pathway is following very, very closely the magnification, whereas the on pathway is doing more a combination of the focus and magnification. In this case, in the average, you see this a little bit lower than that because here you have both the magnification and the focus. We didn't see any evidence, and here, of course, I'm talking about light and dark, okay? So I'm ignoring all the color stuff that, Johannes have demonstrated beautifully, or David Brainerd. Color can be very, very important to give you the sign of the focus, the chromatic aberration and all of this. But when you are thinking in terms of on and off, what we are learning from this small detour that I just did is that they cannot tell you. They can tell you the magnification, but they cannot tell you the sign of the focus. And then, so now we have two things. We know that with on and off, if I am in the retina, I can probably tell you very well how bright it is. This is what retinas do very well, light adaptation in the retina. They can tell you if something is bright, something is dim. And now from the off, they can tell you also if the size, okay, is being, if you're magnifying the scene or you're minifying. I mean, the off pathway has a very, very strong surround suppression. So in some way, it was nice to see this. More surround suppression than the on pathway is gonna be better at telling you what's the magnification of the statistics of the size that you are seeing. So this is the end of this small detour. Then I'm gonna tell you a little bit about visual motor, and then probably I will leave it at that. We are working on this hypothesis, okay? Working hypothesis, could be wrong, that what's happening actually, what is shaping the eye is something very simple, and it's just how bright and how magnified is the image. In some ways similar to this projector that you have there. You have to decide what's the distance of the projector from the screen. In our eye, we have to decide what's the distance from the retina to the pupil. If you put the projector very far, then you get a very large image. You can see your letters very well, but the image gets dimmer. Okay? If you put it very close, you get a very bright image, but the details are smaller. So there is an optimal place where you can get best image brightness and the best image size. And we have a model that we think to work in this working prediction that maybe it's not about the plane of focus, that this is an idea that is out there that the eye stops when it's at the plane of focus. Maybe it's just something as simple as seeing how bright the image is and what is the average size of the grain of the image. And I have some ideas of why I don't like the plane of the focus, but I'm gonna move on. I have a picture of my host here, and I wanted to put this picture just to remind me and to remind the audience that the problem is much more complicated of what I said. And Johannes have been working on these things that I have learned actually from your invitation. I started reading your papers and with more detail. And there are really, really interesting stuff about the contribution of color. And I love the current biology. I recommend to everybody on monovision. This audience is pretty young, but if you get at some point in this thing that comes after 40 that is called presbyopia, I recommend reading this paper to know where to put your far contact lens, if it is on the right side, the left side, and it depends, we are in the US and the UK. Let me go very briefly, over my visual motor, and my time is running out. This is a paper that actually is impressed there, but this is wrong. I was hoping to be impressed by that time. It should be minor revisions. That is the editorial, the official acceptance is not here yet, but is there. So what we did, we became very interested in visual motor aspect. That we started with the pupil, but now we are thinking that, eye muscles, eye position, maybe proprioception. I keep thinking about Michael of these associative things that you can measure in the human brain. But for now, I'm going to just tell you about what they call the triad. Okay, this is the triad in the literature. When you focus something, when something gets close to you, there are three things that change in your eye. First, your lens changes because you have to bring the image to focus. So the lens becomes more powerful. Your pupil constricts. Look at the pupil, it's coming back. pupil constricts, and the eyes converge to make sure that you are there. Hamed is a very bright student in my lab, discovered this thing. I will tell you what discover means after. So the eye that you see turning here, okay, is occluded. Okay, so we have one eye occluded. We have Toby glasses, so we are getting pictures of the eye, but this eye is occluded, the one that is turning, and the other is looking at the monitor. And then we have optotune lens. So what we can do is we put a -5 diopters, and if you are young enough, you cannot do this with people like me. If you are young enough, you can do +5 in your lens, and then what the optometrist calls, you clear the image. So you correct the blur. So we give you -5, but you see it clear. But in the moment that you see clear, when you made your lens to go to +5, the eye turns as if the object was approaching the observer. That is what it would happen if you were doing +5. And the two pupils constrict. So just think about this. This guy, the guy that is moving, is occluded, is seeing nothing. And when we put -5, the focus, what you see is that the two pupils constrict and this eye turns nasally. Okay, so now we are going to measure with light. We're going to put light and dark stimuli. We're going to do the same thing. On and off, how is the on and off affected with this? And these are just 3 slides, and then I'm pretty much done. So the first thing we find that is cool, is that contrast is what matters here, not brightness, contrast. As you make the contrast higher, it doesn't matter that it's a light contrast or it's a dark contrast. Well, it does matter, but for both, for light and dark stimuli, the accommodative eye burden, the amount of turning from the eye increases as I increase contrast. And the pupil also increases as I increase contrast. So as I make something darker, okay, that the pupil eye reflex should make the pupil larger, but no, because I'm talking about accommodative pupil constriction, not pupil eye reflex. The pupil is getting smaller. In this case, the on is stronger than the off because, the pupil eye reflex is very on-dominated. But both increase in contrast. There are some interesting things that I'm going to skip about the contrast sensitivity, but I will just leave it at that. The most important point that I want to make is that if you keep doing this time and time and time, so remember, they do one trial, and then they go another trial. So we can just plot time from 0 to 20 minutes until you finish the task. And what happens is that your eye is turning more and more nasally. For me, the most important thing is that your pupil is getting smaller and smaller. And if you are a myope, that is in green here, this is even much more pronounced. The important thing that is happening here is that when you are recruiting accommodation, and this, you don't need to do this with our fancy -5 lens, you do this every time that you are reading. When you are reading for 20 minutes, your pupil is going to start getting smaller and smaller. And if you are a myope, it's going to be more pronounced. So I have one thing about this. Let me just summarize one thing here. The important thing that is coming from this, okay, is that if you are a myope, you are getting the worst of both worlds. When you are outdoors, your pupil eye reflex is weakened because the on pathway is weakened. So you are not going to get brightness. That is what helps. And on top of that, if you are reading, particularly if you are reading under low light, your pupil accommodative response now overrules light. Light doesn't matter. I want to get sharpness, and it's going to get even more constricted. And then you're going to get poor retinal illumination. This goes back to my model. If you just think about retinal illumination as the problem, this could be doing it. If you read a lot under low light, you're going to get very poor retinal illumination. That could be creating the problem. And if you go outdoors and you already have the pupil, pupil eye reflex disrupted, this could make it worse. Just one very brief thing, 2 very brief things, and then I'm done. Part of this paper, okay, the paper that is accepted but is not accepted yet, we found something cool with blinks. and I just cannot stop from showing you this. We found a U-shaped relationship between blink duration and the pupil constriction. And blink duration, we talk about very long blink durations, up to 2. Basically, our subjects could close the eyes for 2.5 seconds. until they started the next trial. And because of this, we were able to see this U-shaped relationship. And then we did it in single subjects. And in singles, that is what I'm showing you here. Single subjects, basically what we are asking subjects, just close your eyes briefly, okay? Just count one and then open. Now, I'm gonna count 1, 2, and you have to close while I say 1, 2, 1, 2. And now I'm gonna count 1 Mississippi, and you have to close it for 1 Mississippi. And what you see is as you increase blink duration, for short blinks, less than 1.5 seconds, the pupil constriction gets weaker. But as you start 1.5 seconds, you make it longer and longer, the pupil constriction becomes stronger. What is happening? When this is an example here. When you have a very short blink, the pupillary light reflex, that it would be right here, is not strong enough to trigger it, but the accommodative reflex is very strong. And the accommodative reflex, as I show you, is mediated by on and off. On and off. So you are getting a lot of accommodative reflex, but when you are making the blink duration longer, then you are getting the pupil reflex, because the on pathway is stronger the longer the dark period preceding the on response. I'm gonna just tell you one more thing. I'm gonna skip this. Tell you one more thing. Believe me, this is the last thing. This is unpublished. But basically what we're doing is looking, can we figure out, okay, what is happening with on and off pathway vision in the periphery and in the upper visual field? Is the upper visual field the same as the lower visual field in myopia, or you are getting deficits that are different in the upper and the lower? And for this, we are using head-mounted visual displays, similar to what I showed you before. And this is one of my favorite slides. And this is my last data slide. Basically, what we are plotting is in the x-axis is polar angle. So basically, we start here. If I am in my right eye, okay, I start in the horizontal meridian in the nasal, and I go, this is 0, and I go in a circle, go all the way to 360. And I can do this at a radial eccentricity of 5, a small circle, 10, 15, 20, all the way to 25. And you see many beautiful things. The first thing that you see is, what Marisa Carrasco has been talking forever about, these asymmetries between vertical meridian, horizontal meridian, upper and lower field. If you are in the horizontal meridian, so it would be at 0 and 180, you make less errors than if you are in the vertical meridian. That would be at 90 and 270. So basically, if I have a target falling here, I'm going to detect it. I'm going to be worse at detecting it than if it's falling in my horizontal meridian. Not a big deal. We know this. This is a magnification that we have in V1. You just have better vision in humans for horizontal than the vertical. The other thing that you see is in the upper field. So upper field is shown in blue, like the sky, lower field in green, like the grass. And you see that we make more errors in the upper field than the lower field. Everybody does that. The beauty is that now we can see it for lights and darks and with a beautiful precision. So now we can go to 10, you see still this bimodal. We can go to 15, you have the optic disc, the blind spot right there disrupting the entire thing. We can go to 20, and as you get to 25, instead of bimodal, it becomes unimodal. I can explain you why, but what is so cool is that you start making more errors for light and dark stimuli. And there is a correlation with myopia. These are 18 subjects here, and most of them myopes, and there's a correlation. So as you become myopic, you are seeing your periphery. I mean, we have evidence that you are losing, you have peripheral deficits, and you have upper visual field deficits. So on top of not being able to look at the sky because of this pupil stuff that I told you that is messed up, on top of that, when you look at it, you are actually able to see worse. So there is really not a good reason for you to be looking up. The other beauty of this slide is we have measurements of retinal ganglion cell layer thickness here at the bottom for all these subjects. And these two, I thought that it was just way too many plots, but in the paper, you will be able to see the correlations. Basically, you plot these 16 values against these 16 values, and there is a correlation of very, very strong and very significant at all eccentricities. So with this, I'm gonna get to I need some minutes for I'm gonna go skip directly to my lab, because this is the most important slide, and I just want to make sure that I devote enough time to this. All this work was possible, obviously, because of all the funding from different agencies, including NIH, particularly grateful to the National Eye Institute. And all these amazing people. I mean, Jing has been with me, for many, many years. He's a senior scientist in the lab. And basically every single paper you will see his name, he's involved in everything. Mitch Dool is the clinician in the lab. Now, I mean, in the clinician is a clinical professor. I cannot do anything without him now because we are doing a lot of stuff in humans. Jens Kremko, he has his own lab now in Magdeburg. He's a professor there. And he did all the cortical stuff on and off. Reese just got his Saro one in Emory. He did all the on-off asymmetries. Carmen Pons is doing neurosurgery at the University of Chicago, combining research, neurosurgery, and the family. I don't know how she's doing all of this, but she was involved into the magnification, the focus. Saurabh Najafian is doing a postdoc with March Livingstone. The theory of cortical malformation came from him. Hamed Rahimi, really a very, very smart guy. He won actually the best thesis dissertation in the entire SUNY system across the 64 campuses and all disciplines of science within SUNY. And he's now with Winrich Fraywell and doing a postdoc. Sabina Paudel, and he was the guy with the image, the importance of luminance, image brightness in changing contrast sensitivity of on and off. Sabina Paudel is now doing a postdoc, also a PhD student doing a postdoc with Susana Marcos. She was the one that did the ERG for full field and saw the difference of on and off in myopia. Farzaneh just graduated, did the binocular stuff that I love it, and I didn't have time to talk about this. But she was also involved in this last paper with Urusha, that is a current graduate student, this last paper on the visual motor function in myopia, and showing that myopes have a very powerful, potentiated accommodative pupil constriction. And is 101, I will leave it at that. Thank you so much for coming and for your attention.
Question. Can you go into more detail on so my pupillary reflex is messed up by the time I open? Then I go outside and think I get more light than when people see me, but you're saying there's some reflex where you painful or something you avoid looking at the high luminance areas, or how does that?
So the way we are thinking, okay, and thank you for the question because you give me the opportunity to, the way the myopia field thinks, okay, is that myopia was induced by raviola and Wiesel, just by closing the eye. Okay, you close the eye grows. But then Frank Schaeffer in Germany found something really, really cool. Raviola and Wiesel was in macaques, but Frank Schaeffer was in chicken. And what he found is that if you put a positive lens in front of a chick, the eye shortens. Actually, you were talking about the choroids gets thicker, the eye shortens. The eye becomes hyperope. And if you put a negative lens, the eye becomes myopic. So it becomes, the thin choroids, and so everybody started to think about two different mechanisms: one, visual deprivation myopia, or lens-induced myopia. And the idea that is out there, and there's a multi-billion-dollar industry in this, of myopia control, that is based on this idea that the plane of focus is different, and then you put plus lenses to bring the plane of focus to the right place. I have a very hard time believing this because getting the plane of focus is really difficult. I mean, in kids, it takes 500 milliseconds. In adults, it takes 250 milliseconds. The entire brain is involved in accommodation. I mean, from the brainstem, the frontal cortex, cerebellum, you name it. The retina, the way we think about it is that the retina knows something very well. Light. Knows if there is light or if there is dark, how bright the image is. There is entire circuitry just devoted to light adaptation. And we think also that they can do a very good job in telling me the size of the stimulant because there are very small receptive fields. So I should optimize the grain of my image in order to get the strongest response. If you think from this perspective, let's leave the size for now. Let's just think about retinal illumination. Imagine that everything is about retinal illumination. So if you get your retina well illuminated, your eye stops If you get your retina poorly illuminated, the eye keeps crying, to make, to basically to optimize the retinal response. Based on this, being outdoors is good. Why is it good being outdoors? Because it's much, much brighter than indoors. But it's not going to be good if, like you are like my son, my older son, for example, that during recess, he would take his book and there, get in a shady spot, has a lot of myopia, and read for hours. This is not what recess is for. Recess is for, looking up. So if your behavior, so it's in some ways this slide that skipped, it's a combination of visual behavior. This is what we are doing now, measuring visual behavior, measuring thing. You need a bright environment to prevent it. If your visual behavior, intervenes even when you are in a bright environment, you are going to get in trouble. If when you are reading indoors, okay, you have a very bright light so that you get enough retinal illumination, you're going to be fine. But if you like many, I'm meeting now people, real people that have the problem, that love to turn off the lights and then read for the phone, and it looks awfully bright, but it's not. This is just your retina doing what the retina does. That is light adaptation. You feel that you are having a very bright stimulus, but you are having a very dim stimulus. And on top of that, your pupil is constricting because it's overriding the light. It's just caring about acuity. And then you're getting a very poor retinal illumination. And if you do this for a long time, you're going to get myopia.
Could I make you not leave the image size behind? I mean, that's the part of this I'm not quite following.
Yeah.
If to the extent that the images of the world are scale invariant, magnify it, you're going to get the same statistical properties. So where do you think that the signal to the visual system about image size, what the optimal image size would actually be coming from?
The way I see it is that imagine you start with a checkerboard, okay? And then you make this checkerboard, and obviously I'm simplifying tremendously the nature, but just like a thinking exercise. A checkerboard, and now I start increasing the number of checks, okay, the spatial frequency, call it what you want. So I make more and more and more checks to a point that is gray, okay, you cannot see anything else there. Your retinal array is doing exactly the same thing. There is a point here that is not getting any response because there is no contrast. The reflective fields are not small enough to see this small grain. So they are going to get to a point. If I get here, they are too large. Surround suppression, I'm not getting enough bang. There is an optimal size for this checkerboard. And this is where I stay. And when you grow the eye, what you are doing is that you are doing two things. You are doing exactly as you are doing when you are putting the projector farther and farther from the screen. As you grow the eye, you make the image larger, you increase the magnification of the image, but you make the image dimmer. So we think that and of course, there is a combination of brightness and size that will activate the best your retina. And we think that when you reach to that point, this is when you stop growing.
More to talk about.
Yeah.
Okay. So any other questions? All right, well, let's thank the speaker.
Thank you.